bottom of page 134, classification of neurons. And this is it seems a little weird, but it's a great place to put this short little topic because we've been talking about the anatomy of the cells of the nervous system, of nervous tissue, the difference between neuroglia and neurons and the basic structure of neurons. But we're about to get to talk about the physiology and behavior and activity of neurons and what they do and how they do it. So this is a great topic because neurons are actually the cells that do the activity of the nervous system, and they're supported by those all-important glial cells. But neurons can be classified in two different ways. They can be classified by structure, and they can be classified by function. Now, the function classifications that we'll get to in a sec are using the same words we've already been using since the start of this chapter last week, the primary words being sensory and motor, and there's kind of a third classification I'll talk about in a second as well. So that's classifying them according to what they do. All right? But I drew these up here too to help us think about the classification based on structure also. So if you take a look at each one of these neurons, the way that you tell what kind of structural classification, what kind of structure it is you look to the soma or the cell body first. Remember from last week we mentioned this, that's determined by the location of the nucleus and most of the organelles. So here's the soma, here's the soma, here's the soma. The structural classification is based on how many extensions are coming off of the soma or the cell body. Actually, let's start at the very bottom. Here's the soma, and there's one little stem. I know I kind of drew it with a double line, but I wanted to make it really big. I feel like all the textbooks sometimes show pictures where it's not, and I'll show you the picture that's from the Martini textbook in a second. And it's really tiny, so I wanted to accentuate this. This neuron down at the bottom is classified by the fact there is one thing extending from the cell body. Now that one thing splits in two directions, but the actual cell body just has one thing. So therefore it's classified as unipolar, or unipolar. I've heard it pronounced unipolar or unipolar. One pole, one stick, basically. Okay? This next one, here's its soma in the middle, and it has two things extending from its soma, so it's referred to as bipolar. Okay, so it's got two things coming off of its nucleus. And this one up here, these actually come in a variety. There's not really any set number that they have. They typically have multi, the word for lots, multipolar. Okay, so that's kind of how these are classified structurally. That's how they're classified structurally. Right. So the structural classification is based on how many things are. Whoops, I was trying to connect my trackpad. There we go. Multipolar. How many things are coming off the soma, right? There's those classifications, multipolar, bipolar, unipolar, all right? Something that is important to know, all of these, no matter how many things are coming off of their cell bodies, all have one axon, okay? So one axon, everybody has one axon. That's because now we need to talk about the function of these neurons. The axon, I always remembered A, axon away, A, axon away. The axon is going to carry the signal that we're just about to talk about, called an impulse. It's going to carry the signal away from that soma. Okay? For the bipolar, it also has one axon that carries the impulse away. So if it has an impulse carrying it away, this is all one directional. It's all one direction going away from the soma is the axon, right? Now this one in the middle may also have impulses coming towards the soma, but that's not an axon. This is a single dendrite. Okay, this one up here at the top. Lots of dendrites. All of these extensions are all dendrites, and they will all send impulses toward the soma. That's what makes them dendrites, is the direction they carry the impulse. Okay? Now this one down at the bottom, it's got a weird structure. It doesn't really have true dendrites. It's almost like the entire thing is all one big axon that just kind of blows, the impulse just blows right past the little stem with the cell body. But that cell body is still controlling the whole neuron. So it still has a single axon. So that's kind of an important concept. All neurons all have one axon, no matter how many dendrites they have. Or in the case of the unipolar, it doesn't really even have a true dendrite, technically speaking. We might call these dendrites. But that's not usually the term that neurologists use. It's kind of the receptive area. It goes by a lot of different names. Anyway, these are the structural classifications, right? Now, the functional classifications are really based on the direction that these cells carry an impulse relative to the central nervous system. So here's a much better set of illustrations from your textbook. But man, I do love drawing them. And I want to kind of illustrate to you guys that drawing and sketching things does not require you being an artist. I'm a crappy artist, clearly. But it's so helpful. It's a helpful study technique. And you can see it doesn't take more than a couple of minutes to really put something together. So check this one in the middle out. This is what I was saying. There's its soma, and this little stem is so tiny it's hard to see that there's one thing coming out of the soma, because it almost looks like there's two things going two directions. But it isn't. It's an actual stem. And you can see the whole labeling here. This book does, okay, so it does call those dendrites. Okay. It calls these dendrites. I've just seen this in other textbooks, not refer to them as true dendrites. But the whole rest of the extension here is an axon, and it's a true axon. I mentioned this last week, that a true axon has a kind of a consistent diameter. It's a tiny microscopic tube, but it's the same diameter its whole length. Dendrites tend to kind of taper off. They're a little wider, closer to the soma, and they get real skinny towards the edge. So we got a bipolar neuron with its one dendrite and one axon. That means there's two things coming out of the cell body, bipolar. We got the unipolar with its single extension coming out of the cell body, unipolar. And then the multipolar, this one's got 1, 2, 3, 4, 5 dendrites with their little dendritic branches. These are all just considered individual dendrites. And it's one and only one axon. Right? The classification functionally has to do with the direction that they will send impulses relative to the central nervous system. So this goes back to what we talked about first time last week. Sensory is any kind of neuron. It can be any one of these, bipolar, unipolar, or multipolar. But if it sends, they all send signals in one direction. And if it's sending its signal towards the central nervous system, it's called a sensory neuron. If it sends it away from the CNS, it's referred to as a motor neuron. We call it a motor neuron. If it's going out of the brain or out of the spinal cord, it's called a motor. And then those that are within the central nervous system that don't carry impulses into or out of the CNS, they're entirely within the CNS. they just connect to each other within the CNS. They're referred to as integrative or associative, as it says at the top of page 135. Integrative or associative. This is a concept, and you can see at the top of 135, this one is not really in bold, because that's a concept that we're going to talk about a lot in the next chapter after exam 3. We're going to get into talking about the central nervous system. Okay? So what I want to do now is take a zoomed in view of a very, very close up view. Whoops, hold on. Hang on a second. Nope. Sorry, everybody. Technical issues. So I want to go back to my drawing of my neuron that I had before, if this thing will let me. Okay. And just draw my neuron with its soma and its tapering dendrites. And as you saw in that textbook illustration, these dendrites have all kinds of little branches, but they get skinnier the further away they get from the soma. And then there's this thing right here called the axon hillock. And then the rest is called the axon, and it's cylindrical all the way out. All right, so that's we'll look at all the rest in a minute. We are going to zoom in. You like my magnifying lens? There we go. We're going to zoom in to a super close look of that line right there. Well, this is a cell. This is a cell. And what is always around the outside of a cell that forms the boundary? Somebody type into the chat for me. What does this line represent when we draw cells on our page? This is the boundary between the inside of the cell. Yeah, it's the membrane. Now, somebody tell me what that membrane is actually made of. I'm just trying to kind of refresh your memory. Always try to squeeze in a little bit of preparation for the final exam so that we don't forget all the stuff that we've forgotten for the past few weeks. What is that membrane made of? What is a cell membrane or it's called a plasma membrane made of? Phospholipid. Yeah. How many phospholipids? Thousands and thousands. How are the phospholipids arranged? There are proteins as well, but it's mostly phospholipid. A double layer of phospholipids. Okay, so we're going to zoom into this area right here, and remember that I'm going to try to see if I can do a quick sketch of it down here. Remember that phospholipids have heads that do mix with water and tails that do not. Somebody remind me of the word that we use for molecules that do not like water. In fact, they are water-fearing. That's not the word for it, but that is the explanation of why. They are non-polar, which gives them the property of being hydrophobic. So these little lines I'm drawing are the hydrophobic tails. And they are, Tristica, they are hydrophobic because they are non-polar molecules. Okay? So the non-polar tells us about the chemical nature. Hydrophobic tells us about their behavior because of their chemical nature, how they act towards water. How about the phospholipid heads that do like water? In fact, they love water, and their name, the term is they love water, hydrophilic. So they arrange themselves like this in a phospholipid bilayer. And we're zooming into this little tight space right here on this membrane right here to remind ourselves of the arrangement. And that means the way that they arrange themselves is the environment outside the cell is mostly water and the environment inside the cell is mostly water. But the fact of the matter is we want to focus on two other substances floating around out there as well. Should I do my drawing the way that it is in your lecture notes at the top of 135 or the way that it is in the textbook? And I'll explain why I'm debating on this. I'll do it the way that it is in the lecture notes. All right. So we're actually looking, let me switch it up a bit. We're zooming into this part of the membrane right here, not that part. I'll explain why. Yeah, whatever simplifies the process for your learning. And I'll explain why. There's a really important thing we got to understand at this point about the inside and the outside. So towards the top of my phospholipid bilayer will be representing the inside of the cell, the inside of this axon. We're just looking at this long piece of axon. And down here will be the outside, okay? And it turns out that the outside has, whoops, that was weird, way more sodium ions outside than there are inside. There are sodium in there, but there's just way more. Lots more. Way more than three. Millions more. Lots more. I see what you're trying to say, though. I see what you're trying to do. Yeah, you're trying to get ahead of me is what you're trying to do. Inside has way more potassium than outside. Okay? The outside does have some potassium, but inside there's way more potassium than the inside has way more potassium, higher potassium concentration. Now, this is what we refer to as the, and why is this? Why is there so much more? Heba is hinting at what I want you guys to remember about why this membrane is like this. Why is there so much more sodium outside than in? And why is there so much more potassium inside than out? Who does this? Who's in charge of this in the membrane? It's actually embedded in the membrane. The sodium-potassium pump. That's right. Draw a sodium-potassium pump over here. Okay? Now, that sodium-potassium pump, it's embedded in the membrane all along the surface of the membrane. Remember, every cell of your body even has it. I mean, it's not just these I'm not just talking about these neurons. Okay? But the sodium-potassium pump pumps sodium out and potassium in. And every time it pumps, this is what Heba was kind of getting at, it pumps 3 sodiums out, and at the same time, essentially, is going to pump potassiums in, but only 2 potassiums in. Just 2. Okay? Now, guys, you don't need to know this, but that's per pump. And these pumps work about 10,000 times per second. Each pump can do what I've illustrated on the screen about 10,000 times a second, every tick of the clock. So that means in 1 second, the pump can actually pump. It's very efficient. It's crazy. That means the real numbers are in 1 second, it can pump about 20,000 potassiums in, while at the same time pumping about 30,000 sodiums out, which is a difference of how many in 1 second. That's a difference of how many ions. It's an actual difference. I want somebody to actually do the math for me. This one's an easy one. It's an actual difference of 10,000 ions different. Okay? And again, you don't need to know that, but I want you to get a scale of the fact that when we first talk about these sodium pumps, we're like, it pumps 3 sodiums out and 2 potassiums in. Why is that such a big deal? It seems like a very big deal. Yeah, every second it's 10,000 more out than in. in 1 second. Okay, you're about to find out. You're about to find out the answer to that question. Every time that it does this, what the point of all of this is there's going to be way more positives outside cell than inside. So we often refer to the inside of the cell as negative and the outside as positive. Neither is exactly true. We're just saying one is more positive relative to the other. Okay? Now, I think Tanya is answering Tristika's question. What's the purpose for the imbalance? And I said I'd be getting to this. But I would say this. We refer to this, as you can see at the top of page 135, we refer to the state as the resting membrane potential. But I ask you this. Who's resting? Certainly not that sodium-potassium pump and its thousands and thousands of other sodium-potassium pumps all on this one cell. And the full name of the sodium-potassium pump is it's called the sodium-potassium ATPase. Why is that its full name? What does it cost? What does it cost? ATP. It costs energy. 10,000 ATP per second per pump. And each cell has thousands of these pumps. So I mentioned this a few weeks ago when we first talked about the sodium-potassium pumps, and I'm going to mention it again. It's estimated that over 50% of the energy and calories that you consume every day actually gets spent on sodium-potassium pumps. It gets converted into multiple different forms, but eventually when it's turned into ATP, over 50% of your daily ATP expenditure is spent just on sodium-potassium pumps. All right? So, I argue that resting membrane potential is kind of a misnomer. These cells are not sleeping at all. They work very hard to maintain this state, but I'll explain why it's called the resting membrane potential. Okay? So, I'm going to get back over here to my actual diagram from the textbook. And the diagram from the textbook, I think, is going to show the membrane flipped the other direction. So the outside is going to be towards the top. The inside is going to be toward the bottom. All right? So hopefully you're ready to kind of flip this. I've drawn it the way that it is at the top of page 135. Okay? And by the way, before I clear my screen on this, the actual charge difference between the outside and the inside can be measured in units called voltage. Volts is just a way of measuring the difference in charge from outside to the inside. And for cells, it's usually around 71 thousandths of a volt, called 70 millivolts. The reason why we use the negative symbol is we always measure it relative to the inside. And so what we're saying with negative 70 millivolt is we're saying that the membrane is 70 millivolts more negative inside than outside. But the reason for that is this constantly working pump, the constantly working pump. Now, hopefully you remember us talking about this from back in muscle. I mentioned it, but a lot of this is going to kind of sound familiar. So here's that same diagram from make sure I'm on my right slide. Yeah, there we go. From the textbook. So this is the outside. You can see extracellular fluid. Here's the inside, the cytosol. You can see the charges. There are positives outside and there are inside. So when we measure it with a little voltmeter, we say the inside is measuring negative 70 more negative than the outside. And all that's saying is there's just a charge difference. Okay, and we just kind of let this voltmeter sit here, and it stays pretty stable at negative 70. And we say the cell is at rest. It's slightly more positive outside, slightly negative inside. What causes this? In my questions, we've already answered. But what causes this is our sodium potassium pump, sodium potassium ATPase? Yes, it definitely requires a lot. And this condition is still, despite the fact that it requires ATP, still known as the resting membrane potential. Okay? And the weird thing that happens is if you kind of mess with the membrane or bump or move the cell, this experiment was first done, so I'm going to say, I'll try to do the math really quickly, like 140 years ago, back in the 1880s, and somebody had a little voltmeter, and they were measuring the difference in the cells and the inside to the outside of the cell. And it was just, the cell was just like this. It was just like humming along, -70. But when they pushed on the cell with a probe, with a little micro probe, kind of bumped it, if they bumped the cell, check it out. It just kind of -70, but they bumped the cell and it kind of goes like that. The needle went up like that. It became less negative. Like that. It doesn't make that noise, but every time they bumped it, they noticed it became less negative. And the scientists kept experimenting with it. And if they bumped it hard enough, they noticed the needle would temporarily go all the way up above 0. What that means is measuring the inside of the cell becoming positive inside temporarily. All right, so that's what we refer to as what's known as an action potential. It's a reversal of the resting potential. Okay, so if you look at what we got just mentioned at the bottom of page 136, this is what happens kind of from the inside of the cell perspective. Happens from the inside of the cell is the inside. I'm going to have to draw that membrane again. I'm going to do it quickly. Here's the phospholipid heads on either side. And with that little probe sticking into the inside becomes positive. Whereas the outside is now temporarily negative. So this is kind of pictured, like I said, at the top. I said bottom of page 136, but there's actually a little diagram. Oh shoot! Hold on a second, guys. I'm gonna flip my picture so that it matches what's in the lecture notes. Because I know not everybody has the textbook, but you've all got a copy of the lecture notes. So here's the membrane again. Just say, can I do it like that from now on? So this was the inside of the cell, where it was mostly potassium, but it was more negative. And there was potassium ions, and there's potassium out here too, but there's a lot more potassium out here. Okay. And then here's the outside where there's way more sodium. Again, there's some sodium in here too, but way more sodium. And there's lots more sodium outside than there is potassium inside. Okay, so this is the membrane at rest. All right, so that's the membrane at rest. But when you mess with the membrane, something happened to it, and scientists were looking at this going, what is happening? And what was happening was some of the positives came inside, making the inside temporarily reversed, positive inside. All right? And so they would measure it like this. Okay? Check it out. I'm gonna kind of skip to, I gotta clear this up. I'm gonna skip to the bottom of 136. To try to reconstruct this for you. Make it make sense, I hope. Okay. So on this graph, what we're looking at is over time, over a span of a few milliseconds, okay, over time, and voltage, millivolts, okay, right there at that spot is right down here. We're going to put -70. This is our cell at rest. All right. So if we're looking at that cell with that little probe in it and just measuring the cell at rest, then over time it would just look like this. Just kind of humming along at -70. There it is. And then somebody bumps it with that little probe and it goes like this. It goes like that. It doesn't make that noise again, but that's the noise I imagine. It kind of bumps up. So what happened right here was it became a little less. It got kind of irritated because we were trying to wake it up. A membrane did. It got irritated. And what happened right here? Let's see. It looks like my network signal is messing up a little. I'm going to turn my camera off. You guys should still be able to hear me okay. And hopefully that'll help. Okay. Tell me what happens, what you think happens right here, going up right there, to make the cell become a little bit more positive inside. Well, I can't read it. Yes, sodium comes in. Okay, so what happens at that specific spot, the cell is allowing a little sodium to come in from the outside world. Okay, yeah, now technically the term for it is depolarization. That's right. But I want to make sure that we're doing the right thing again. There is a name for the event, and there's a thing that causes the event. Okay? So it's kind of like what we were talking about before with the neurons. So what happened, if you get asked what happened, it's sodium came in. Some sodium came into the cell. Humans gave a name for that. Humans gave a name to that. Now, Dongsil, this isn't an action potential yet, but humans gave a name to this and called it depolarization. Okay? So it's referred to as depolarization. All right? Now, Dongsil, this is a good point, a great point. I want you all to see why this is not an action potential yet. Have I actually reversed the charges inside the cell? Now, this whole little chart is looking at the inside of the cell, but if you look at my chart, I will not have reversed the charges until my charge inside the cell gets above 0, correct? As of now, the inside of my cell is actual. I can't see the cell. I'm just measuring what's on the voltmeter. But as for now, it's still negative inside. But that doesn't change the fact that Tashai and Iba were both correct, both in what causes it and what its name is. This is a little bit of a depolarization, it's a name for it, caused by sodium coming in. All right? Now, it goes back to rest because that sodium quickly gets pumped back out by our friends, the sodium-potassium pump. So it goes right back down here to the resting line, and it hums along at rest. But if I really irritate the cell enough with enough stimulus, I will wake up that cell from rest and cause it to depolarize so much that so much sodium comes in that now this is still a depolarization, and it's still caused by sodium coming in. All right? But it's such a strong stimulus that it causes all of these sodiums outside to be allowed to come in all at once. Okay? So this is still a depolarization, still caused by sodium coming in. It's just, to use a non-technical term, it's way more sodium than I had over there on the left. Okay, so that's called a depolarization. Then, here's what they also observed. It quickly, almost instantly, drops back down like that. Okay, now, what term did they give to that when it drops back down? What did they call it? When it's going back down there, when it's going back down to negative. The term they used for it was a repolarization. Yeah. What is it caused by? What causes it? Why does it become negative inside again? No, it's okay. I don't worry about your spelling on the chat. Potassium out. Potassium out. It loses. Potassium goes out. Okay, so potassium goes out, causing. Now remember, potassium is also positive, but when all the positive potassium leaves, it takes its positive outside with it, leaving the inside to become negative again. Okay, so here's the key takeaway. Depolarization because of sodium in, repolarization because of potassium going out. But one happens first, followed by the other. They do not happen at the same time. Okay, so first is depolarization because sodium comes in. The second thing that happens is repolarization because potassium leaves. And the key thing is only if the inside of the cell, yes, kind of getting back to what Dong Sil was mentioning before, only if the inside of the cell actually becomes positive temporarily do we get this reversal of inside charge. It was negative at rest. It was way below zero at rest. But we let so much sodium in from outside that temporarily the inside becomes positiver. That's not a real word, but it's more positive. Then that triggers the opening of potassium channels, letting potassium rush out. Potassium can't wait to get out now. Potassium's positive too, and it's repelled by all these positives coming in. Positiver. It's a real word. It's a real word to my 4-year-old. Does everybody see it? This is really what all of page 136 is all about. It's all about what that's about. Okay? Now here's some key concepts at the top of the next page too. All right? Before we look at that, has anybody got any questions before we look at some of the concepts about this? About some of the ideas that are important to remember about this. Okay? No questions because your minds are so stunned and blown. You got broke it. I broke it down. I think you said broke it down really well. Yeah. Okay. Yeah. I would say that is probably my job. If you're a teacher, that's probably supposed to be your job. It's harder being a teacher in anatomy and physiology because anatomy and physiology is really hard, and breaking stuff down, it's kind of hard to do, but I feel like I'm pretty good at breaking it down. Break it down. Reminds me of my old school days watching Yo MTV Raps. Break it down. But that was a thing. Breaking it down was a thing that I could not do. Did not mean that I didn't enjoy watching other amazing breakdancers do their stuff. Speaking of breaking it down. Now, so here are some key concepts. Now, this is the way it looks. This is the way that thing looks in your textbook, which also does a great job of breaking the whole thing down. All right. So I know, 20s and the millennials, they don't get it. They don't get it. Sorry, I don't want to get off on this, but millennials have no clue what MTV meant to us and our generation. It was super. It was the core of our existence. It was the center of our life. So look at how the textbook breaks it down. Here's the resting potential at -70. There's our 0 point. Now, this one's can be a little more confusing because there are a lot more details on it, but the details are here. The details are important. So here's our 0 point. I shouldn't talk about millennials. Yeah, that's true. You're right. No offense. Millennials are old enough to remember how important. Anyway, never mind. So I'll talk about some other time when we have more time. I will talk about what there is a single show that led to the ultimate destruction of MTV, and you guys can decide. We'll talk about it after class, and we can see if we agree. 0 point right here. It is not until this line goes above 0 that we have an actual action potential. Up to that point, it's just a little bit of a depolarization, and it does not become a true action potential until the charge hits this point called threshold. Threshold is this point where all of the sodium channels nearby suddenly all open at once. They're called voltage-gated sodium channels. Now, guys, we'll get to that in A&P too. Guys, one place that's definitely confusing, we talked about this in, excuse me, got the hiccups. Talked about this when we talked about muscle before. Thing that can be confusing. Do not confuse the sodium, potassium pumps that are pumping the sodium and the potassium using ATP don't confuse them with sodium channels and separate potassium channels. Oops. Okay, don't confuse these. Sodium and potassium channels are just doors. They're like doors in the membrane that will let either one of these ions rush through. They're just doors that open to let the ions go down their concentration gradient from high concentration to low concentration. The actual concentration gradient is set up and created by the pumps. The pumps that are pumping all that sodium out are making it so the sodiums desperately want to get in. Okay, and that's, if you want to just go back to page 135, in the middle of 135, Ms. Long, Dr. Schultz, but also Ms. Long, who co-wrote this, really brilliant kind of, it's not in bold, but it's a great description. There's approximately 35 times, I'm in the middle of 135, approximately 35 times more potassium inside the cell than outside the cell. Potassium would like to leave the cell, but the negatively charged proteins hold it in the cell. And also, it's a charged ion, so it can't cross that phospholipid bilayer. Okay? Next, there is approximately 10 times more sodium outside the cell than inside the cell. Sodium would like to move into the cell because of its concentration gradient, but the membrane is impermeable to sodium. All right? Because the sodium pumps keep pumping those sodiums out and potassiums in. Sodium would love to come in, but can't until, check it out, until the inside of the cell reaches this called threshold that literally causes all the sodium doors to open. They're called voltage-gated sodium channels, but it helps to think of them as these microscopic little doors in the membrane that just bing, they just open all at once, and sodium goes from high concentration to low concentration, comes gushing in. As the sodium comes in, the cell depolarizes, is another word for it. The membrane depolarizes. All right, up here at the peak, where it's at positive 30, that causes the opening of potassium channels. So the potassium doors open. One thing to notice, these channels are highly specific. Those sodium doors that open only let sodium move. They will not let potassium out. Sodium only comes in because it's going down its concentration gradient from high concentration to low concentration. Okay? So at this peak up here, the potassium gates open and potassium goes out, taking its positive out with it, leaving the inside to become more negative, to repolarize again. Okay? Okay. Then there's this leftover period where the sodium channels are closed. The potassium channels are closed, but there's still a little more potassium leaking out, where it's much more negative inside. So there's this whole time period that kind of goes in two phases, known as, I've got it over here on the left. Now we're back to the top of page 137, known as the refractory period. The refractory period. Okay? If you're a parent, are the channels only? No, this is a neuron. This is an axon. This is an axon. These are in the axons of the neuron. Yeah. They're the same as the ones that we talked about in the muscle. They're the same as the ones we talked about in muscle, but they're on axons too. Yes, absolutely. And you guys, I am really getting back to trying to answer. I'm going to skim back through the chat. Way earlier, when Tristica, yep, it was at 2:53, and she typed into the chat, "What is the purpose for this imbalance?" I am still trying to answer that one question while actually working through the notes, because what Tanya's question helps highlight is many tissues. You're welcome. Many tissues of the body have these sodium channels, and the pumps set up the resting potential that allow the cells to use channels of various types to do work, to send signals, to transmit impulses, to communicate with each other. This is how the cells of your body communicate long distance with each other. Okay? So we are currently not talking about muscle, but it is the same. Does it make sense? Yes. Good. So it's the same thing found on the surface of the muscle that's also in these axons. Now, we haven't even gotten to the good part yet. I think this stuff is exciting enough, but it gets better. Okay? It gets more fascinating. So hold on. Buckle your belts. We're going to go for a big ride. So there's a period in this whole process where the cell becomes positive inside, and then it repolarizes. And there's a whole time period here where it is in the middle of something. Okay? If you have children or have ever been a child and had parents, you have heard a parent say to you, "Hold on, please, I am in the middle of something." And it's so weird as a kid. You think, my parents are so mean. I'm never going to be a mean parent like my parents were. Well, just you wait and see is all I've got to say as a parent. So I think of this refractory period as the neuron says, "Would you please hold on? I'm in the middle of something. I am in the middle of an action potential." And this time period where the neuron can't do another action potential because it's in the middle of an action potential, and it doesn't matter how hard you stimulate the cell. It's in the middle of finishing an action potential over here. And if you will just wait a second, I'm sure you never heard your mom say this, but she said something that sounds like this tone of voice. If you will just wait a millisecond, I will reset my ions and I will be right with you. I love you, Mom. I miss you. If your mom is still alive, send her a love. Send her some love. Call her this afternoon. Text her. My mom passed away 13 years ago from kidney cancer. And still miss how that woman would talk to me like that. Man, she would be. No, it's okay. I don't mention it. Thank you. I don't mention it for that, but I mention it to say, after class today, send mom some love. All right? So, yes, I will. I will help you when I am good and done with my action potential. I am in the middle of an action potential right now. That is called the refractory period. The neuron cannot do another action potential because it's either in the middle of one, that's called absolute refractory period. It's right in the middle of an action potential. Or the relative refractory period, which is a little bit after that, which kind of means it could do another action potential, but the stimulus would have to be stronger. It'd have to have a much stronger stimulus for that relative refractory period. It doesn't look like at the top of page 137, you need to know the difference between the two types of refractory period. I think it just talks about that absolute refractory period that's happening during it says during depolarization, but it really, during depolarization and a little bit of the repolarization. Okay, another key concept, all or none. All or none means that once this cell has gotten to this threshold point, the charge will go all the way to the peak and have to come all the way back down to reset. The concept is like this. If you don't get to the threshold, I'm going to have to erase all my cool little lines. If you don't get to the threshold, it's like that little bump that I drew on the previous slide. You can get a little bit of a bump, but if you don't get to threshold, it immediately goes right back to rest. But if you do get to threshold, then you're going to get a full action potential. It doesn't matter how strong the stimulus was. That's kind of the idea. Once the threshold is reached, the action potential is on. It is on. Oh, it's on. Okay, it's going to go full out to the peak, and it's going to have to reset. Okay, another key concept is the idea that this action potential that we're looking at in this diagram might be happening at a specific spot on the axon. But that action potential that I've got an arrow pointed to right there can then become the stimulus for an action potential there, which becomes a stimulus for the action potential there, which becomes a stimulus for the action potential there. You see where I'm going with this? It causes a chain reaction of action potentials all along the axon. So the point of this is, all you need to do is start an action potential at that axon hillock. And once you get an action potential at that hillock, then you will get this kind of jumping action potentials all along the length of the axon. And this is how axons communicate long distance. This is what we refer to as, it's called saltatory conduction. It sounds like the word salt, but it literally comes from the Latin word that means jumping or bouncing from node to node. And it can travel fast. This makes it travel fast. And it jumps from node to node because of that myelination caused by the Schwann cells. So that's why myelinated neurons can carry action potentials a lot faster, because the action potentials jump from node to node without having to fill all the space in between. Okay? And that actually contributes to the speed. This is kind of under letter D, middle of page 137. The fastest neurons have myelin. There are neurons that are unmyelinated. It's true that there are some that have no myelin. They just send signals a lot slower, which I said last week is okay. There are places in a completely normal nervous system where we need that. Speed is not always the most important thing. Sometimes it's good to slow down, slow your neurons down and think about things for a while. In fact, a lot of things that we refer to as thought and decision making involve a lot of unmyelinated axons very close together working on a problem or memory or something like that. The other thing that adds to the speed is the diameter of the axon. So if you kind of look at this axon, and it's pictured way down here in the bottom right, and you kind of measure its distance across, lots of axons come in a variety of different diameters. There's some that can be a larger tube and they are faster, and some that are smaller tubes and they are smaller. That it's not really a tube, it's just actually a tubular cell, tubular extension of the cell. So A fibers are those that are the largest diameter, very, very high speed, 100 meters per second. Okay, so just in case you're not familiar with the metric system, let me put it this way. If you're not familiar with the metric system, you're probably like me and you grew up in America, which means you are familiar with American football. So I'll give you an idea. 100 meters is not exactly, but it's close to 300. It's close to 300 feet. It's kind of close to 100 yards. It's not far off of a football field. So if you could imagine a runner being able to run from one end of the football field to the other, including the end zones, and be able to do that in 1 second, that's how fast these neurons send impulses. Considering the fact that you're not even 100 meters tall, most of us are not even 2 meters tall. If we do it in meters, I'm probably, I'm 5'10", so I don't know how tall that is in meters. I think that's 1.7 or 1.8 meters or something like that. So I have neurons that go from my brain to my spinal cord that can send impulses from brain to spinal cord in way less than a second, about 1/100th of a second. Those are my really high-speed A fibers. The C fibers are the much slower ones. The B fibers also have myelin, but these are smaller in diameter, and so I think these tend to be a little bit slower. So those concepts are really good concepts in understanding how these action potentials are transmitted from the beginning of the axon, at the axon hillicus where they start, and then they shoot all the way down the length of the axon. Now, here's the key question. What happens? Any questions about this before I switch it up a little? I'm gonna switch to another concept. Let's go back to drawing again. Drawing is always a good idea. Let's draw an axon again. Let's draw our soma. There. Here's our axon. And it's going to have an action potential that starts right there, that stimulates an action potential there, right? And so what we're doing is we're actually sending a series of action potentials. I always picture this, imagine this is like tipping over dominoes, something I used to love doing. My grandparents and my great-grandparents, every Sunday night, we would get together at my grandparents' house, and the kids would play while the grandparents and the great-grandparents played dominoes. And so there were always boxes of dominoes around. And as a kid, I loved setting up and knocking over dominoes. And so I don't know if that's anything you've done, but it's definitely something. There are videos you can look up on YouTube. So you can kind of check that out. Domino videos is what my kids, one of my kids especially, loves to watch them. And I imagine this is like tipping over dominoes. One tips over the next one, and the tips over the next one, and tips over the next one. Okay? Now, Heba, this is a good question right here. This is a good question right here. Heba asked, what is hyperpolarization for? What is the hyperpolarization for? So the hyperpolarization is during that refractory period. And if we look at this axon, if we look at, well, hold on, I'm not done. I'm not done yet. This is a good concept because it's related to exactly what I'm talking about right now. So your timing is perfect. If I look at an action, if I look at an action potential happening right here, remember what that means. If the cell is having an action potential at that spot, it means that sodium is rushing in. It's depolarizing. It's becoming positive inside. But that becomes the stimulus to trigger a new action potential right here. Why does it not cause a stimulus for a new action potential back here? because going backwards, this is where my analogy of the dominoes, that domino has already been toppled over. It needs to be set up again. All right? So this action potential on this spot, let me kind of label it real big. We're going to talk about an action potential right there at that spot. This action potential happening here, yes, it is all or none, but it's also the stimulus for an action potential over here on the other side of a Schwann cell. And remember that I'm skipping the Schwann cells and going to the next node. So this action potential becomes the stimulus for a new action potential here because this part of the membrane over here is at rest. It's resting potential. But this action potential cannot stimulate an action potential back here because this membrane is not at rest. This part of the membrane, this part of the axon, is in refractory period because of hyperpolarization. This part of the membrane is in the middle of hyperpolarization, which means it hasn't reset all of its ions yet. It's still hyperpolarized. It's overpolarized. It's over negative inside. It's extra negative inside. That's what hyperpolarization means. It's extra negative inside. Okay? So where an action potential could stimulate the nearby areas of the membrane, it can only do it out here where the membrane is actually at rest, not back here where the membrane is hyperpolarized. So what does that mean about the direction of the impulse? Tell me what that means about the direction of the impulse. What I've got graphed up here, these bouncing little arrows, is the impulse traveling. Why is it only traveling that way? Not necessarily descending. Descending only if we're talking about, it's always only one way, is my point. One way. And it's away from what? Always away from the what? It's on my diagram. Always going, those arrows are going away from the soma. Yeah, always away from the soma. So the thing that carries the impulse away from the soma, these bouncing action potentials away from the soma, is the axon. The axon, AA, axon away. Okay? Now, okay, Tanya, it would be descending if, I'm going to draw a little brain up here. Let's draw a wrinkly little brain. It would be descending if the axon was up here in the brain. If the soma was up here in the brain and its axon was going down the spinal cord. True. But it would still be one way if the cell body was here in the spinal cord and going out to a muscle, which would not technically be descending. All right, so that's why, your comment about descending only is not, it's only descending if the cell body is in the brain and the axon is in the spinal cord. Okay? The concept that I'm trying to communicate that's kind of challenging is to get back and make sure everybody gets, Heba has had a good question. What is the role of that hyperpolarization, right? I've explained it. Now let me give you a straightforward answer. I did it backwards. I should have just given you a straightforward answer and then only explained it if you had a question. So I've explained it. Here's a straightforward answer. The straightforward answer is to make sure the impulse travels only one way, away from the axon hillock. Nope. The straightforward answer, the purpose of the hyperpolarization is to ensure the impulse only travels one way, away from the soma. The purpose is not more negative. The function is not more negative. That's exactly right, Tanya. That's it. It's a great analogy. And in fact, if you guys look in the chat, Tanya says it would be like a bullet fired from a gun, the bullet firing from the chamber through the barrel of the gun is exactly right. Now, let me ask you this, Tanya, since you brought it up, but anybody can answer this. Can I make the bullet travel faster by squeezing the trigger harder?
No.
By squeezing the trigger harder. And again, not everybody knows how a gun works, so that's fine. If you don't know how a gun works, the analogy doesn't work, unfortunately. Can I make the bullet travel really slow if I just squeeze the trigger gently?
No.
Can I make the bullet travel, 3 or 4 miles an hour by just gently squeezing the trigger?
No.
Okay? So a gun works by all or none. As soon as the trigger activates the hammer and the hammer hits the back of the bullet, I no longer have any choice about where that bullet is going and how fast it will travel there. This is the concept of all or none. And this is the reason why, I don't always like to use the analogy because, like I said, a lot of people don't know how a gun works. So that I don't like using analogies that I prefer not to use analogies that don't make sense to everybody. But a lot of textbooks use this analogy. A lot of textbooks I've seen in A&P will use this analogy. It's like firing a bullet from a gun. Once you trigger the hammer, the bullet will fire at a set speed determined by the bullet, the amount of gunpowder in the shell, all kinds of things, but none of them are in your control anymore. Very much the same way that a series of action potentials will travel only one way, away from the soma. See how I swung this back around to our concept? And that traveling action potential is what's known as an impulse. It's what we call an impulse. It's actually a series of separate events. It's one domino hits the next domino. And each individual domino has exactly one job to hit the next domino. And that's it. It's once it hits that next domino, job over. So if you could watch a video of dominoes tipping over in slow-mo, you'd see each domino becomes the stimulus for the next domino to fall, which then becomes the stimulus for the next domino, and so on. Okay? But once you tip over that first domino, done deal. Anybody that's ever played with dominoes knows how frustrating it can be to set them up, because if you touch one the wrong way, you've tipped over your whole set. So this is how that all or none idea works. But let's get back to the bottom of page 137. Okay, what happens when this impulse gets down here to the end? My question is, where is it going? Let's zoom in to this little area right down here and kind of zoom in. And what we see is the axon kind of enlarges and has this sort of flat surface at the end. And it gets to an end. And at the end of this, now we've met this before. Good. Well, hey, whatever analogy worked, I think put them both together in your head. I'm glad they work. We've seen this before because the end of this axon, called the axon terminal, is filled vesicles. And those vesicles contain chemicals. So when the impulse gets all the way to the end of the axon, okay, now what we're going to see here is sodium in, depolarization, causes potassium out, repolarization, but that becomes the stimulus for this area to let its sodium in and its potassium out. Depolarization, repolarization, and action potential, basically. Those two things together make what we call an action potential, dipole, repole. Okay? But that becomes the stimulus for another action potential. And then when we get right down here, we don't have sodium channels anymore. We don't even really have potassium channels. What do we have that we saw in the muscle chapter? We have channels for what down here? Let's see who remembers. This is reviewing for next week's test too. Calcium channels, yeah. Down here we got calcium. Calcium channels. And the calcium stimulates these vesicles to move over to the edge of the end of the cell and merge with the end of the cell, spilling their contents. These chemicals that are inside. But is that it? Exocytosis, exactly. But is it neurotransmitter? I'm being kind of coy. I'm being kind of silly. Hang tight. This chemical is only neurotransmitter if there's what over here? What do we got over here? Now in the muscle, what was over here? What was over here in muscle? This whole area. There's a lot of things over here, but the whole area was referred to as the MEP. Yeah, sarcolemma, but a specific area of the sarcolemma called the MEP. Who remembers what that's called? The motor end plate. Oh, Dungsil, you type that fast. The motor end plate. That includes, Heba, includes receptors. Receptors for what? Receptors for these chemicals that got spilled out up here because calcium came in, because calcium channels opened, because the action potential triggered the opening of calcium channels. But guys, we only have that. Here's my point. We only have that if there's a cell, another cell over here on the other side with its phospholipid bilayer that I just tried to represent with a single line. That's the phospholipid bilayer right there. We only have that if there's a cell on the other side. And we have a cell on the other side of this space. Then we have what? What do we have if we have one cell communicating across a space? Stop drawing on my drawing. Communicating across the space. I will let you come to the whiteboard in a few minutes if you want. That's fine. What do we have if we have one cell communicating across the space to another cell? Oh no, now I know it's you. Bigger. Bigger, Tanya. Bigger. Bigger than synaptic cleft. Synaptic cleft is the name for the space. But if we have one cell communicating with another cell across that cleft, what's the whole arrangement called? Anybody. I'm not just picking on Tanya. What's the whole thing called? No, not a motor unit. It's only a motor unit if this over here on the other side is a muscle. I am not assuming this is muscle. We have talked about this being muscle, but this, in the example we're about to use, this doesn't have to be a muscle. It does not have to be a muscle. Guys, it's in capital letters, all caps at the bottom of page 137. I'll wait. I'll wait for you to see it. It's the heading. Yeah. There, you found it. Let me say it again. No. That's okay. I was giving you the definition before I was giving you the word. All right, so let's flip it around. What I was describing was a synapse. No, not a synaptic cleft. Okay, make sure you guys get this. My definition was not a part of the synapse. I was talking about a cell communicating across a space with another cell. That is called a synapse. The space between them is called the synaptic cleft, but that's only the name for the synaptic cleft. It's only the name for the synaptic cleft. So let me walk it back one more time. Okay? Be patient with me. A cell communicating with another cell across a space is called a synapse. The space itself is the synaptic cleft. It's the cleft or the space of the synapse. The first cell that's doing the communicating is referred to as, top of page 138. The cell that's doing the communicating is called the presynaptic cell, and it communicates across the cleft to the postsynaptic cell. Okay? I'm going to try to make this as straightforward as I can, because there's a lot of stuff that you can read about there on page 138, but I'm going to break it down to the bare minimum. Okay? The presynaptic cell is the presynaptic cell because it's the one that releases these chemicals. The presynaptic cell is the presynaptic cell because it's the one that actually releases the chemicals. It doesn't have to be acetylcholine. It doesn't have to be acetylcholine, and they're not necessarily neurotransmitters. I'm going to define that for you. Hold on, I'm getting to it. Don't worry, I've done this before. I'm leading you to it. We're getting there. Just follow me along the yellow brick road. We'll get there. We won't get lost in the field of poppies. Okay? The presynaptic cell is the presynaptic cell because it releases chemicals. Okay? The postsynaptic cell is the postsynaptic cell because it has receptors for those chemicals. This cell is the postsynaptic cell because in the membrane, on the other side, there are receptors that double as channels. Now, I know why everybody's wanting to say acetylcholine because that was the first one that we ever talked about a couple of weeks ago when we talked about how a neuron activates a muscle. It's true. It's true. It goes back to page, I'm going to go back to it again. It was that page with the diagram on it. 112. Goes back to 112, right? Okay. But what I'm saying is that is only one example of a synapse where the presynaptic cell is an axon terminal. Yes, Dong Sil. Where the presynaptic cell is an axon terminal in that case. And on page 112, the postsynaptic cell is a muscle. But the postsynaptic cell doesn't have to be a muscle. In fact, most often, it's another neuron. And this is how neurons communicate with other neurons across these little spaces. One releases chemicals that the other can receive, and it changes the membrane of the other to make the membrane either more positive inside or more negative inside. It makes some kind of change over here in the postsynaptic cell. If the chemical that's released by this first cell has receptors on the second cell, if the chemical released by the presynaptic cell has receptors on the postsynaptic cell that can do something to that postsynaptic cell, then we refer to that chemical as a neurotransmitter. A neurotransmitter is a chemical specifically released by one cell that causes a change in the charge, the voltage of the membrane on the other side of that space. Okay? That's what makes this one a presynaptic. It has chemicals that it can release. That's what makes this one the postsynaptic. It has receptors that can receive the chemical, the neurotransmitter, and when it clicks on the postsynaptic cell, it makes the postsynaptic cell a little more positive or a little more negative on the inside by letting sodium in, was the example we talked about on page 112. All right? So do you so you think, are you guys starting to get the connection with 112, page 112 with muscle, but the muscle isn't how it always is. Muscle is not how it always has to be. That was just the first example that we ever talked about. Okay? So let me go back to my slides to use a figure from the textbook again, is it not always calcium that gets let in on the presynaptic side? As far as I know. As far as I know, I will be on the presynaptic side. On the presynaptic side, not calcium on the postsynaptic side. So on that now these schematic diagrams are kind of flipped differently. So we've got that presynaptic cell here at the top. This little lightning bolt, the little red lightning bolt, is the action potential, sodium in, potassium out, sodium in, potassium out. And it opens calcium channels on this cell, which causes the cell to release a chemical across the space. And on the other side of the space, there are receptors that bind to the chemical. And when that chemical binds, it opens sodium channels, which makes the inside of the cell more positive. Okay, so I'm trying to say the same words that I've been saying, kind of putting it all in the same context, what I've been saying. But helping you kind of look this slightly more technical version with more details from the textbook, but it's only sort of saying the same thing that I've been saying with some added details. Okay, so this cell up here, this is the end of this is just the end of that axon. The end of that long distance impulse that's been coming from the brain or spinal cord up there somewhere. Or this could even be happening inside the brain spinal cord. How one neuron, this is the axon terminal. Axon terminal of a neuron, and this could just be another neuron. Okay. Oh man, that's just a mess. I'm sorry. But this could easily just be another neuron. The only example we talked about prior to this was if this thing down here at the bottom was when this thing down here at the bottom was the muscle, the surface of it was the neuromuscular junction. So this was the motor end plate. These were acetylcholine receptors, and when the acetylcholine binds, it opens the channel to let sodium in, and that causes an action potential across the surface of the muscle. That's true. But my point is, what I really want to emphasize is, this is how all neurons communicate with each other. This is really how all neurons communicate from neuron to neuron. This is kind of the whole point of a neuron sending an action potential. It's going to get to a point where it gets to the end. But just because it's the end for that neuron doesn't mean it can't communicate to another neuron and get that neuron either more excited or less excited. Okay? So, that's what makes this cell presynaptic. It's the one that releases the chemical. That's what makes this one down here at the bottom postsynaptic. It's got receptors for that chemical. Okay? And when it's a neuron communicating to another cell, we broadly refer to that chemical as a neurotransmitter. That's where we use the word neurotransmitter. We call it a neurotransmitter if it's used by one neuron to communicate with another cell. This is my second heart today. I'm on a roll. I must be explaining this stuff pretty well for some of you guys. Okay? Good. Leave a little something in the tip jar. Just kidding. Joking. I was waiting for that. Butter, straight butter. So that's where we get these names now. Okay, so as we move from 138 to 139, we're going to take a break in just a few minutes, but I want to kind of get through all of this neurotransmitter stuff so that your brains can cool down during a short break. Okay, so stick with me. As we move from the bottom of 138 and then to the top of 139, these are kind of connected. There's a couple of paragraphs at the bottom of 138 that are talking about acetylcholine, but it connects with what's at the top of 139 because, let me kind of center it before I change the screen. What you can see happening down here in this image, acetylcholine binds to the receptor on the other side, which opens a sodium channel. Now you can see it may be really tiny on your screen, but sodium comes in, which will do what to the charge inside the cell? What's that going to do to the charge inside this postsynaptic cell? It's going to make it more what? Sodium coming in is going to make it more something or less. It depends on how you phrase it. So we become a little more positive. Yeah, a little more positive. Yeah. So if this cell over here, where my little pointer is, if it was at rest, it might become a little bit more positive until it reaches threshold. If enough sodium comes in, this cell could reach threshold and then get itself its own action potential going, which is pictured right over here in the very bottom right. Propagation of an action potential if generated. That sounds so technical, but that's what it does. It might get enough sodium in that can make the inside of the cell just positive enough that we get this thing happening. An actual action potential. Remember, it's got to jump above 0. But we've got to get enough sodium coming in to get a depolarization going and opening all of those channels. So this is what's happening on this postsynaptic cell here. If it does that, if the neurotransmitter allows this postsynaptic cell to get a little depolarized, it allows it to get just a little bit more excited. So that's why we call that an excitatory postsynaptic potential. It's still a potential because it's not a full action potential yet. It's just making it a little bit more excited, more likely to be able to get an action potential. So that's why we refer to acetylcholine as an excitatory neurotransmitter. It's an excitatory neurotransmitter because it makes the inside of the cell a little less negative, more likely to reach its threshold. And it kind of would look like this on if we were to graph it. It's not going to be a full action potential. Here's that threshold point that we saw on the previous diagram. I kind of drew it, I kind of repeated the diagram right up here. But there it is, drawn as a dashed line on the top diagram. They just didn't include it up here. But it means that if a neurotransmitter makes that postsynaptic cell, the one that was at the bottom of that previous picture, makes it a little more excited, then this kind of becomes its new starting point rather than here. This becomes its new starting point means this is much closer to threshold, making it far easier to fire an action potential. So excitatory neurotransmitters make the postsynaptic cell a little more likely to reach threshold, and that they just slightly depolarize. They slightly depolarize that postsynaptic membrane. Okay? But some neurotransmitters can have the opposite effect. So now, this is not acetylcholine, but there are some neurotransmitters that might have the opposite effect. They might make the inside of the cell actually more negative. These can work a lot of different ways. But what is one way you think that you could make the inside of the cell a little more negative at rest? What kind of channels could you open that would make the inside more negative? Potassium. Yeah. Some neurotransmitters actually open potassium channels, which lets some potassium out. Remember, potassium wants out, and it's positive, and when it goes out, it makes the inside a little more negative. So some neurotransmitters can actually make it harder for the cell to reach threshold. And that is a hyperpolarization. So these are hyperpolarizing neurotransmitters that we refer to as inhibitory neurotransmitters. Okay, another way that you might not have read about, another way of doing it is actually opening chloride channels. Chloride ions, and chloride ions are negative, and that lets chloride ions into the cell, making the inside of the cell much more negative. Now, when would you want to do this in a clinical setting? When would this be something that we could actually use with a pharmaceutical drug, with a pharmaceutical agent? How about Novocaine? Anybody ever had a shot of Novocaine? Sure, giving birth. Novocaine is a drug that actually blocks sodium channels and allows some chloride into the nerves around your teeth. And it hyperpolarizes the pain receptors. Well, it hyperpolarizes really all of the nerves in your jaw. It hyperpolarizes them, making it almost impossible for them to reach threshold, making it almost impossible for them to communicate pain impulses to your brain while the dentist is drilling your teeth. Epidural works in a similar fashion around the neurons, around the spinal cord. Yep. We also have a drug that works kind of the same, that we use usually for topical administration. Lidocaine. You can actually buy little tubes of this stuff. Lidocaine. And we have a drug that is very similar that we use in spray form, a mist form. You might have heard of it. All in the same family: cocaine, lidocaine, Novocaine. And they all kind of hyperpolarize neurons on the surface. And cocaine is used often when you're doing a laryngoscope or a gastroenteroscopy. It's true. It is solubilized so that it only lands on the surface, but it deadens the gag reflex. If the doctor needs to look down in your voice box, in your larynx, or needs to go down and look inside your stomach. I don't know, Tristica. I really don't. It's best if we just don't talk about that. Yeah. If you have had that, yeah. if you've had that done before, then under the direction of a doctor or technician, then it is not illegal. This is a controlled substance, which means it's controlled by doctors, nurses, and people with the key to the cabinet, right? But that's kind of how these drugs work. They cause inhibitory, inhibitory postsynaptic potentials that make it harder for these neurons to send impulses. A couple other topics, then we'll take a short break. The way that nerves work together. So what I mentioned before was we were looking at a very close-up view of this spot right here on your screen, guys. This was an axon terminal from another axon. There's the presynaptic cell, and this whole blue thing over here is the postsynaptic cell. And what's happening between, there's a little gap there called the synapse, and we're zoomed out now. We're not looking quite as close. But this cell is releasing neurotransmitter that is causing this postsynaptic cell to depolarize a little bit. So it's a little bit of an excitatory action potential. But that may not do anything because it may not add up to enough of a stimulus here in what's called the trigger zone. This is the axon hillock. Your textbook calls it the initial segment, but our lecture notes kind of refer to this as the trigger zone. This trigger zone, much like with the analogy of the gun, it needs to reach its threshold to be able to fire a full action potential. This little stimulus is not quite enough to do it, but that neuron can send a quick second stimulus that could help this trigger zone release its action potential, and then you get a full impulse traveling. That's called an action potential propagation in the textbook, and we just refer to a traveling impulse. An action potential here becomes the stimulus for an action potential here, and so on, all the way down the axon. Okay? If it does that, if this first neuron sends a second stimulus quickly in a short period of time, that's called temporal summation. The word summation just means adding on to it. Summation, adding on. Temporal means it's adding another one in a short time. The word temporal referring to time. Okay? So that axon hillock is going to add up any of those potentials, and if it's enough, the hillock will propagate an action potential. The other kind of summation is if there is another neuron. Check this out. I don't know if you guys can read it. The text is small in this caption. If there's another neuron close by, that's what's called spatial. Spatial means nearby or close. So if there's an action potential releasing neurotransmitter from one neuron, and then a separate neuron also adds its opinion, its excitatory neurotransmitter, and they're close enough to each other and close enough to the hillock, then that axon hillock will add those two signals together at the same time in the same location, and that's referred to as spatial summation. And to be honest, both of these are usually at work. Don't think of temporal and spatial summation as really separate things. They're just two different ways that one axon, in this case the purple one, gets the second axon, the blue one, to fire an action potential. And if it can't get that second one to fire an action potential, it'll send a quick impulse to follow it up, or it might recruit a friend to help it. That's spatial summation. And if it all adds up to that negative 55 millivolt threshold, that's when you get blue neuron firing its action potential all the way along. Okay. So common neurotransmitters that can do a lot of these things are acetylcholine. We talked about a lot and mentioned already that it is excitatory. And remember what that means is it's excitatory, which means it causes an excitatory postsynaptic potential, which means it slightly depolarizes the postsynaptic cell, slightly depolarizes, makes it a little bit more likely to reach threshold. And the major place we have seen it since a couple of weeks ago was the neuromuscular junction of skeletal muscles that all somatic motor neurons use acetylcholine. That's not the only place it's used. Acetylcholine was the first ever discovered neurotransmitter. I got to give a shout out to Sir Henry Gale, British researcher who actually won the Nobel Prize back in like the 1930s for his discovery of acetylcholine. Part of the reason why it was the first one discovered is because it's so abundant. It's used so abundantly throughout the nervous system. It's really common. So certainly not the last time we're going to talk about acetylcholine. We're going to talk about the autonomic nervous system. I guess about 3 weeks we'll talk about the autonomic nervous system, and we will talk about acetylcholine again. We will talk about it again in A&P 2 when we talk about the effects of acetylcholine on the heart, blood vessels, so on and so forth. It can be used as a drug that you give a patient to slow their heart rate if they have tachycardia, all kinds of things. So by a long shot, this will not be your last encounter with acetylcholine. So that's one good reason for mentioning it first. Remember that it's broken down by cholinesterase. We talked about that with the muscle before. There are drugs we have that can interfere with this mechanism on purpose to try to control the use of the mechanism of acetylcholine. There are some neurotransmitters that are just straight up amino acids. Glutamate is an example of one that's used commonly in the central nervous system, in the brain primarily, and GABA. And know this one, guys. Know this one. How many stars does this one need? GABA. You don't need to know what it stands for. I'm going to see if I can remember. Gamma aminobutyric acid. I'm going to stop adding stars, but the reason why it's so important to know is it's one of the few well-known inhibitory neurotransmitters, and inhibitory in this case is not a bad thing. It's a balance. It's keeping the other excitatory neurotransmitters balanced out, and we need inhibitory neurotransmitters. Let me give you a quick example. Sorry to kind of get sidetracked a little bit, but a quick example. If you guys have ever heard of the disease called lockjaw, lockjaw goes by the medical term tetanus, and tetanus is caused by a bacterial toxin. The name of the bacterium is Clostridium tetani, and it produces a toxin called tetanospasmin. And the toxin it crosses the blood-brain barrier, so it can get past those astrocytes, and it gets into the spinal cord, and it blocks the release of GABA, meaning without GABA, you lose your inhibitory neurotransmitter, so motor neurons don't know when to stop sending impulses to muscle, and they won't stop sending impulses to muscle, so your muscles will contract but cannot relax. GABA is critical in the central nervous system in telling motor neurons to stop sending impulses so that muscle can relax. Okay, now there's also another example in your lecture notes. This Valium is something that increases levels of GABA to kind of relax. Valium, I think, it's gotten a bad rap, but originally it was kind of a muscle relaxant. Was how it was originally marketed and produced back in like the 1950s, 1960s. But it also reduces anxiety, and it's a very, very useful drug for all the negative press that it gets. Here's another one we are going to talk about a ton this semester and next. Norepinephrine. In fact, norepinephrine is a very important excitatory neurotransmitter in the autonomic nervous system, and we are going to see this theme a lot, both in a couple of weeks and throughout A&P2, because you are going to learn in your programs, not just nursing programs, you are going to learn so many medications in pharmacology, in your pharmacology classes, that mess with the balance between acetylcholine and norepinephrine receptors on the cells of your body. And the balance between the two, you're going to be able to learn chemicals and drugs that will manipulate that balance. Just kidding. Dopamine and serotonin. Dopamine can be inhibitory or excitatory. In the central nervous system, it is an important excitatory neurotransmitter, but it is an inhibitory neurotransmitter when it comes to regulating muscle contractions, and not necessarily making them stop, but making them smoother. In fact, the lack of dopamine being produced by a part of the brain called the substantia nigra is a part of the brainstem that produces a lot of dopamine that helps to regulate muscle contractions. In the absence of it, you develop a tremor and a disease known as Parkinson's disease. So you can't control muscle contraction, and the muscles develop this shake known as the Parkinson's tremor. which is not a great name for it, but it's an old school name for it. So dopamine actually is an excitatory neurotransmitter with kind of an inhibitory effect. It's hard to explain, and we don't bother explaining it in your lecture notes because it is kind of complicated for now, but you'll study it maybe at the next level. Serotonin is very important neurotransmitter in the GI tract. Serotonin enhances smooth muscles that help move food through the GI tract, so it really enhances digestion. It also improves or increases appetite. It elevates mood, and it gives you a sense of ease and reduces anxiety to assist in sleep. It's not the only one that we'll talk about that helps in sleep. We'll study a few others more in A&P2 that help with regulating sleep, but serotonin has a role there. There's a few others that are mentioned, gases like nitric oxide that relax smooth muscle, endorphins and enkephalins that actually can tamp down pain, and so on. But we'll take a break there. We're going to flip back to page